Hey, hi everyone. So just before we get started with our invited speaker for today, I just wanted to make a quick announcement. We've been asked by the Science Undergraduate Student Society to promote an event that's coming up next week, so on November 6th. That's the Wednesday, I believe. And it is an opportunity for science students to learn about possible future careers. So although you are in your first year, for most of you, never too early to start thinking about this, and you might be trying to decide on what major to pursue in your second year. So please feel free to attend this event if you want to find out more about possible future career opportunities. So I'll just leave that open for a moment if you want to scan the QR code, and then we'll hand it over, I think, to Marcia for an introduction.
Excellent. Yeah, thank you, Caitlin. Hi, everyone. Welcome. Happy Halloween. Welcome to the Sci 103 Science and Society lecture series. We have a real treat for you today with our guest scientist, who I'll introduce in a moment. Of course, just a few reminders about the speaker series. It's great to see many of you joining our webinar live on the heels of what I'm sure has been a busy midterm season. And I'm sure you're all working very hard on your term papers as well for Sci 113. For all students, whether live with us today while watching the seminar, definitely take note of some of those questions that you might want to ask today's guest scientist. And you can use the chat function or the raise hand function. We hope that at the end of today's seminar, there'll be time for our speaker to answer any questions that you may have. But just, of course, as a reminder that this is an academic setting and to ensure your questions and comments are respectful and on task. As a reminder, too, that while it's important to complete 4 out of 6 of the speaker series worksheets over the course of the term, we really want you to be watching the talks, or for those of you here live, to be taking note and to be prepared for next class to debrief the seminar. And also just as a reminder that the topics presented will be relevant for your final projects at the end of the term. With that, it's my absolute pleasure to introduce our speaker for today, Dr. Selena Sagan. Dr. Sagan is a professor in the Department of Microbiology and Immunology and one of my colleagues here at UBC. She completed her PhD at the University of Ottawa, trained as a postdoctoral researcher at Stanford, and is recognized as a Canada Research Chair in RNA biology and viral infections. Her research team focuses on viruses that pose major health threats around the world, viruses such as hepatitis C virus, dengue, and Zika virus. She's also leading a multi-million-dollar research effort to help us be better prepared for pandemics in the future. Her work uses state-of-the-art tools to study how molecules like RNA and proteins interact inside host cells during viral infections, with the hope that understanding these sorts of hijacking of viruses inside our cells and how viruses reproduce and spread, Dr. Sagan and her team can better understand the diseases that they can cause. So thank you again, Dr. Sagan, for joining us, and I'm excited to hear about your work.
Thank you so much. I'm really excited to share some of our work today. So let me just share my screen. And then, how's that looking?
Looks great.
Okay, great. Awesome. So I'm just going to minimize this so that I can go through the presentation. So thanks so much for the invitation to speak today. I'm really excited to tell you a little bit about one of the projects in our lab. And today I'm going to tell you about microRNA management of viral infection. But before I start into some of the science, I want to tell you a little bit about myself and my path in science. So as Marcia kindly introduced me, I'm a professor in the Department of Microbiology and Immunology in the Faculty of Science here at UBC. I'm a Canada Research Chair in RNA Biology and Viral Infections. I also am the co-director of the Prepare for Pandemics Through Advanced Research and Evolution Research Cluster here at UBC, which is a hiring cluster of professors that are interested in pandemic preparedness. I'm also the president of the Canadian Society for Virology, that I just took over this position this past summer. What do I do as a faculty at UBC? Some of you might be wondering, what else do we do other than teach? Well, primarily I really run a research lab, and I run a research lab that studies the role of RNA at the host virus interface. And you can feel free to check out our website if you're interested in the topics that I talked to you about today. And most of my job is actually supervising graduate students, postdoctoral fellows, undergraduates, and research staff. I also serve on thesis committees. And with these students, we really spend most of our time brainstorming, hypothesizing, designing, and doing experiments to try to figure out how things work, and in particular, how viruses work. I also write papers and I apply for funding by writing grants to support the research that my lab does. I participate in peer review of grants and papers. I give research talks at different conferences and universities, and I teach in courses like this, as well as some other courses at UBC at the second and the fourth year level. And of course, I sit on a lot of different committees at the department, university, or research community level. And so I perform sort of service. And so these are sort of the three roles that professors participate in at universities: research, teaching, and service. So how did I get here? I want to tell you a little bit about my path scientifically, about the things that interested me along the way and how I started my lab on the topics that we study. And then I also want to tell you a little bit about precisely where the geography of sort of my training was. So like you, I did an undergraduate degree in science. And I really got excited about science when I started thinking about the central dogma. And so I want to start with the central dogma today. And many of you will know the central dogma, which is that DNA is transcribed into RNA, and RNA is, and this is done by polymerases in our cells, which are the enzymes that copy nucleic acids. And RNA is translated into protein, and protein is what kind of makes up all of our cells. So DNA is our genetic material where we store our genetic information. RNA is a copy of the DNA, sort of the instructions, and then RNA gets translated into protein. And protein is sort of what we think of as like what makes up all the different things in our cells. And proteins are translated from RNA by our host cell ribosomes. And also, DNA has to be replicated in order for our cells to divide and for us to become who we are. And DNA gets replicated by another type of polymerase as well. And I was sort of taught this probably in late high school and also in my undergraduate degree. And when I got into my undergraduate degree and I started learning and in my biochemistry classes, I started to feel kind of like I was lied to. You know, DNA was this storage information, RNA was this messenger that was there just to make protein. But I realized that RNA was more than just the messenger in my biochemistry classes. And this is one of the first moments that really like blew my mind in science, because what I learned was, like DNA, RNA can also store genetic information. So it can also sort of be the library for genetic information. And because RNA is single-stranded, it can fold into all these complex structures, something that DNA can't do. And like proteins, then RNA can actually fold into these complex structures that can actually catalyze chemical reactions, just like proteins do. So it actually turned out that RNA wasn't just the messenger. RNA was the best of both worlds. It could both store genetic information, and it could actually catalyze chemical reactions like proteins could. And so I started to learn about different types of RNAs, that there were RNAs that weren't coding, that weren't messenger RNAs, that just became RNAs. They weren't destined to make proteins. I learned about catalytic RNAs. I learned about microRNAs, genome editing RNAs, and even RNAs that could bestow mortality. So these are RNAs which help to ensure that our telomeres are long enough in cells. And I think the thing that really set this in motion for me was learning about the RNA world hypothesis. And the RNA world hypothesis says that actually maybe RNA predated both DNA and proteins in the world. And maybe RNA is actually the precursor to life on Earth. And so that kind of solidified for me that RNA was more than just the messenger and was this really interesting molecule that I wanted to learn more about. So if we go back to the central dogma, again, I can tell you about the second moment that kind of blew my mind during my undergraduate degree. And that's when I learned that not only could RNA do all these cool things, but RNA could actually be the template for DNA, and RNA could actually be reverse transcribed to make DNA. And not only that, not only does DNA replicate, but there were actually RNAs that could replicate as well. And RNA could be copied into RNA. So it could also be the template for replication. And we learned about these two processes, this reverse transcription and this replication of RNA, from studying viruses. And so viruses became something that I wanted to learn more about. So what is a virus? Well, in fact, viruses can be really, really simple. They can be so simple that they're just a piece of nucleic acid surrounded by a protein coat. And they're really, really, really small, really, really, really small in general. However, even though they're very, very small and very, very simple, they can cause a lot of damage, disease, and devastation around the world. And interestingly, viruses can evolve genetically, but we don't even consider them living organisms or alive. And that was sort of the second moment in my scientific career, which really kind of blew my mind. And so I kind of knew that I was really interested in RNA biology and I was really interested in virus infection. And so I decided that with my scientific career, what I was going to study was RNA viruses. And RNA viruses allows me to study both my loves. So I can study both RNA and viruses, and then I can selfishly study both of my two passions in science. All right. So let me tell you a little bit about my path to the University of British Columbia. So I'm from Ottawa, which is Canada's capital. And I did my undergraduate training at McGill University in microbiology and immunology. And that's sort of where I learned about RNA and viruses. And so when I wanted to do a PhD, I decided to head back to Ottawa and do a PhD in biochemistry, microbiology, and immunology, where I could study RNA and viruses. And then from there, during my PhD degree, I also spent some time at the University of Edinburgh doing a research elective, understanding RNA and RNA structure, which I'll tell you a bit about more in a moment. And then after my PhD, I did a postdoctoral fellowship, which was kind of like an apprenticeship that you do before you become a professor in some cases. And so I did this at Stanford University, where I studied microRNAs and viruses. And then I started my own lab back in 2013 at McGill University in the departments of microbiology, immunology, and biochemistry. But in 2023, so last year, I relocated my research lab to University of British Columbia. And so we've been here for just over a year. So like many of you, I'm new to UBC. So I already told you that my research interests as an undergraduate student at McGill University was in RNA and viruses. And that's what really kind of blew my mind about science. And so when I went to do my PhD, I decided to study RNA and viruses, and I studied RNA viruses. I studied a plant virus, RNA, and I studied a particular protein from this plant virus called P19, which is kind of shown in this image. It's a dimer, so there's two P19 molecules that interact together. And I studied this phenomenon in plants where plants, when they are infected by a virus, they cut up that virus, viral genome into pieces, and then they use it to target the virus as an antiviral response. And so RNA plant viruses have developed strategies to sort of have a counter defense. And this is a counter defense used by plant viruses. They make this protein called P19, and the P19 protein actually binds to these small RNAs that are pieces of the viral genome, and it hides them from the plant so that they can't use them against them. And so this is kind of what I studied during my PhD, so I could study both RNA. I could study RNA viruses and study both my two passions. And during that time, I started to get really interested in RNA structure and how RNAs fold. So I did a research elective at the University of Edinburgh looking at the structure of viral RNAs. And this was a very formative time for me. I learned a lot during my time at the University of Edinburgh working with Peter Simmons. And I learned about this really fascinating phenomenon. And that phenomenon is that RNA viruses, or at least positive sense RNA viruses, they have a lot of structure in their genomes, not just at the ends of genomes, which is how we kind of think about RNA structure, but actually throughout their whole genome, they have lots of structure. It means they can fold into these complex structures. And interestingly, the amount of structure a virus has through its whole genome is actually correlated with its ability to persist in hosts. So viruses that have lots of RNA structure can actually persist or cause chronic infections in their hosts, whereas viruses that don't have a lot of structure are actually like acute or resolving infections in most cases. And this was really fascinating, just that the structure of an RNA could actually determine whether a virus is acute and resolving or becomes chronically infected. And so that kind of blew my mind. I don't think we really understand or we understood then why that was, but I think we're starting to sort of unpack some of that, those phenomenon now. And it's something that our lab is certainly interested in. And then after my PhD, I moved to Stanford University to work with Peter Sarnow. And in Peter's lab, I was working on microRNAs and their interactions with viruses. And microRNAs are another class of small RNAs that are made in our cells. And I'm going to tell you a story related to this today. So when I started my own lab, as I said, I decided to study RNA at the host-virus interface, in particular looking at interactions between RNA viruses and their hosts. And we don't discriminate in the Sagan lab. We study RNAs, whether they're small or large. So we study really small RNAs like microRNAs, which are only about 20 nucleotides in length. And we study really big RNAs like viral RNAs, which can be 10,000, 12,000, 15,000 bases. And I started my lab at McGill in 2013. And then again, I moved my lab to University of British Columbia last year in 2023. And the questions that really drive me as a scientist really revolve around RNA viruses and how they work. And the thing that I'm really fascinated about is that in positive sense RNA viruses, they actually only have a single piece of RNA, one piece of RNA. And that RNA actually has to be a template for everything the virus does. It has to be a template for the ribosome to make the viral proteins, but it also has to be a template for replication, for the polymerase to copy the RNA into the negative strand and then to make more viruses. And it also has to be a template for packaging. So it also has to condense into the viral particle. And so it kind of blew my mind that one piece of RNA could do all of these different things. And the reason it could do all these different things is because it's really dynamic. It folds into all sorts of complex structures to recruit other proteins and RNAs to allow it to go through these different stages in the viral life cycle. So the things that my lab are really interested in are how do viral genomes fold? What regions of the viral genomes are important for mediating the different events in the life cycle? How are these different events regulated? What host and viral factors help mediate these events and how? And of course, how can we interfere with these processes, right? We want to be able to develop antivirals and things that can stop viruses in their tracks so that we can stop disease. And of course, by studying one virus, we can learn a lot about other RNA viruses. We can also learn about how RNAs are regulated in the cell. And so these are all sort of things that really drive the research that I do today. All right. So in the lab, since I've had the lab for about 10 years, we have lots of different research projects. But today I'm really going to focus on this one, the microRNA management of viral infection. All right. All right. So my timing is really good for this lecture because some of you will know that the Nobel Prize in Physiology or Medicine this year was given to my friend Victor Ambros, as well as Gary Revkin, for the discovery of microRNAs. And microRNAs are these really tiny RNAs that have really profound physiological importance. So let me tell you a little bit about microRNAs. So what are microRNAs? Well, they're very small non-coding RNAs. So non-coding means they don't produce any protein. They're very small, so they're only about 21 nucleotides in length, so they're very short. They're very highly evolutionarily conserved. So that means that in mammals, lots of us have the same microRNAs. And so they're conserved across different species. And what they do is they mediate post-transcriptional regulation of gene expression. So what that means is after an RNA is transcribed, they can regulate it. And so a microRNA gets transcribed just like a messenger RNA in our cells. And that microRNA can then come back and it can bind to a messenger RNA and prevent it from making protein. And through this activity, they're actually predicted to regulate about 60% of all human genes. So this is a pretty big discovery, and not surprising why Victor and Gary won the Nobel Prize for this discovery. And because they regulate so many human genes, they're implicated in a lot of different diseases, right? Including cancer, viral infections, cardiovascular, and metabolic diseases. And of course, today I'm going to tell you about an interesting interaction with a virus. So just briefly, let me tell you a little bit about the microRNA pathway. So as I said, microRNAs are transcribed just kind of like messenger RNAs in our cells, and they actually form these hairpin structures, this folded structure, and they get cleaved in the nucleus into these short hairpins. And then they're exported to the cytoplasm where they're further processed into these short microRNA duplexes. So you have two strands here. And this recruits the RNA-induced silencing complex, which contains an Argonaute protein. Argonaute protein is one of the proteins I want you to remember today for the purposes of my talk. What the Argonaute protein does is it takes that microRNA duplex, it unwinds it, and it holds one of those microRNAs, the mature microRNA, inside the Argonaute protein. And then it uses it as a template to find RNAs in the cell that match the sequence of the microRNA. And when it finds a messenger RNA that has the sequence, if it's perfectly complementary, the Argonaute protein cleaves that RNA and it gets sent to be degraded. But in most of our cells, messenger RNAs aren't perfectly complementary to the microRNA. In fact, they have imperfect complementarity. So what happens when they have imperfect complementarity is the Argonaute protein can't cleave the RNA. Instead, it sort of gets stuck there. And usually microRNAs bind to the 3' UTR, so the end of the RNAs in our cells. And when it sits there for a little while, it ends up calling its partner protein, or the microRNA effector protein, called TNRC6. And what TNRC6 does is it induces translational repression. So it means that RNA no longer makes protein. And it also calls degradation enzymes, so deadenylases and decapping enzymes, which would degrade the RNA from the 3' end and then as well from the 5' end. And so that RNA gets degraded. And so this is sort of a simplification of the microRNA pathway, and microRNAs typically downregulate target genes. But today I'm going to tell you about an unusual interaction between a human microRNA and hepatitis C virus. So the microRNA that we're going to talk about today, or my favorite microRNA, is called microRNA 122. It's a evolutionarily conserved liver-specific microRNA. And so you can see on this northern blot that there's lots of this microRNA in the human and rat liver. There's actually about 135,000 copies in every liver cell in our bodies. And you can see it's evolutionary conserved. So you can see the zebrafish is stained with microRNA 122 and its liver is lit up with this microRNA in purple here. And the normal role of this microRNA in our cells is to regulate genes that are involved in cholesterol synthesis and fatty acid metabolism. All right, so I'm going to tell you today about a sort of non-canonical role for a microRNA in the life cycle of hepatitis C virus. So let me tell you what hepatitis C virus is. So hepatitis C virus is an RNA virus. It's a positive sense RNA virus. So the genome contains just one piece of RNA, and I've kind of depicted it here with a very highly structured 5' end. So this is the front end of the RNA, a structured 3' end of the RNA, and then it has an open reading frame in the middle, which encodes 10 proteins. And this is pretty prevalent. So there's about 71 million people worldwide affected by hepatitis C virus infection. It's responsible for about 40 to 60% of chronic liver disease, and it's the leading cause of liver transplant in the world. And of those who become infected, about 85% will become chronically infected. And what I mean by that is they will never clear the virus without antiviral therapy. So they'll have the virus for their lifetime. And this can lead to chronic liver disease and a need for a liver transplant. So there's no vaccine available to prevent hepatitis C virus infection. But we do have very good antivirals today that can cure patients that have chronic hepatitis C virus, but accessibility is certainly still a barrier to treatments in different countries around the world. All right, so what does the life cycle of a virus look like? And I'm just going to very briefly go through this, and you don't need to remember all the details. But briefly, viruses enter cells. In this case, in Hep C, it enters through an endosome. And in the endosome, it uncoats, releasing its genome from the viral particle into the cytoplasm. And hepatitis C virus is a positive single-stranded RNA virus, and it's ribosome ready. So the viral RNA can call the host cell ribosome to the RNA to make the viral proteins immediately upon entering into the cell. And so it calls the ribosome, the RNA gets translated into the viral proteins, and as the viral proteins accumulate, hepatitis C virus sets up replication factories or replication organelles, which are the sites where it replicates its genome. And what it does is it takes that positive strand RNA, it converts it into a negative strand, so it copies it into a negative strand, and then it uses that negative strand to make more positive strand viral RNAs. And then once enough of the viral genomes have been made and a lot of the proteins have been made, they can be packaged into viral particles, which then exit the cell through the secretory pathway and are released from the cell and can seed a new infection. And today we're really going to focus on sort of the translation step and the RNA replication step a little bit today. All right, so I told you that microRNA 122 has an unusual interaction with the HCV genome. So again, here's, I've depicted the HCV genome here, and it has highly structured RNA in the 3' and the 5' UTRs. So 5' is the front, and the 3' is the end of the RNA. And these are involved in replication and assembly of the virus. It also has this structure in the 5' end known as an internal ribosomal entry site, and that constitutes these stem loops here. And the internal ribosomal entry site is a way for the virus to call the ribosome to the RNA. So most of our messenger RNAs in our cells have a cap, and that's how they call the ribosome. But hepatitis C virus doesn't have a cap. Instead, it folds its RNA into a structure which can directly call the ribosome to the RNA. And in the very 5' end, just before the iris structure, there are two sequences which bind to microRNA 122, which is that liver-specific microRNA I told you about. And this is a really unusual interaction for 3 reasons. So the first reason it's unusual is that microRNAs actually normally bind to the 3' end or the end of messenger RNAs, but this one binds right in the 5' terminus. And actually, this is the very 5' end of the viral RNA. The microRNA actually base pairs with the very terminus of the viral genome. The second reason it's really unusual is because there's 2 microRNA 122 sites in very, very close proximity. You can see here they're only actually separated by a single base in the viral RNA. And the third, and probably the most unusual thing about this interaction, is that rather than downregulate gene expression and decay the RNA, like I told you microRNAs normally do in our cells, it actually upregulates hepatitis C virus RNA accumulation. And you can see that really nicely on this northern blot, where what I've done is I've used a locked nucleic acid or an antisense molecule that binds to the microRNA and sequesters the microRNA from the virus. And when you do that, you see you completely lose HCV RNA accumulation in cells. Now, this isn't just true in cell culture, this is also true in human patients. So patients that had chronic hepatitis C virus were treated with an antisense microRNA 122 molecule, so a molecule which binds to our liver-specific microRNA. And they were given a single dose of this microRNA inhibitor, and you can see that they all had dose-dependent and sustained reductions in HCV RNA accumulation. So they were given a single dose, the microRNA levels drop, and so do hepatitis C virus levels. But interestingly, as microRNA levels started to rise in the patients over time, some of them got hepatitis C virus infection back. But interestingly, 2 patients actually cleared the infection just from a single dose of this microRNA inhibitor. And so this microRNA is really, really important for HCV to be able to accumulate in the liver of chronic hepatitis C virus patients. And so when I started my lab, I really wanted to know, oh, how does this microRNA do something so opposite to what it's thought to do? How does it promote HCV RNA accumulation? All right, so one of the things that we started to think about right away was perhaps because the microRNA binds to the very 5' end or the very end of the RNA, maybe it protects the HCV genome from being degraded. And so the idea was that when the microRNA is bound to the genome, it prevents cellular sensors of RNA or pyrophosphatases, which would remove the triphosphate at the end of the RNA, or nucleases, which would degrade the viral RNA, such that when the microRNA isn't there, these could act on the viral RNA and cause them to be decayed. And so that's what we started to investigate. And that was done by my first 2 graduate students, Annie Bernier and Jasmine Shahal, who graduated a few years ago. And so what Annie did was she looked for cellular sensors of viral RNA. And there's a number of different cellular sensors of viral RNA in our cells. There's one called PKR, the RIG-I-like receptors, which are helicases, which are sensors of foreign RNA, as well as the IFIT proteins. I'm going to make a really long story short because Annie looked at all of these different molecules to see if microRNA 122 was protecting the genome from them. But in fact, microRNA 122 wasn't protecting against any of them. So then Annie turned her attention to cellular decay machinery. And so she looked at pyrophosphatases. She found 2 pyrophosphatases in the human genome, one called DOM3Z, and another called DUSP11, which remove a triphosphate from RNAs so that they can be decayed. And she also found a ribonuclease called XRN1, which is what decays RNAs in our cells. And in fact, she found that all of these were involved. So let me show you a little bit of the data. So one of the assays that Annie and Jasmine did was they produced the HCV RNA, just the end of the RNA, just a fragment of it. And then they performed XRN1 assays. So XRN1 is that endonuclease, which chews up RNA. And so they did XRN1 assays in the presence of no microRNA, and you can see that the RNA degrades very quickly when you add XRN1 if the RNA is there alone. Then they did the same thing, but they added a microRNA that doesn't interact with the fragment at the end of the genome. Again, you see that RNA decays quite quickly. However, when they added microRNA 122, which could bind to this fragment of the HCV genome, they noticed that the RNA decayed more slowly. So you can see that these bands take longer to disappear, suggesting that the microRNA could protect the RNA from being degraded. But this was just in vitro. It was just with fragments of RNA. So we also wanted to be able to test this in the context of infection or replication. So how do we study hepatitis C virus replication in the lab? Well, we make use of a reporter virus. And what I mean by reporter virus is we took the viral genome and we placed a luciferase gene in the middle of the viral genome. And then this RNA we can introduce into cells in culture. And what happens is the RNA gets introduced into the cells and it can replicate in those cells. And when it replicates, it produces this luciferase enzyme. And then what we can do is we can do a luciferase assay and look for the cells that produce light. And that serves as a proxy for HCV RNA replication. We also have a defective RNA where we've made a mutation in the RNA-dependent RNA polymerase. We call this GNN. And that mutation actually causes the polymerase to be dead. So it means that the viral RNA can't replicate. It can go into cells, it can be translated, but over time it just degrades because it can't replicate. And so this is what this looks like. So when we do a luciferase assay with wild type viral RNA, so viral RNA that's replication competent. You introduce it into cells, it translates, it replicates, and so you see this curve goes up really nicely as the viral RNA accumulates. However, if you use the GNN, which is replication defective, you can introduce the viral RNA into the cell and it can translate at the beginning to produce luciferase signal, but it only decays over time and quickly it decays below background, which is this dotted line here, because the viral RNA isn't replicating or accumulating. And so this is one of the ways that we study HCV replication in the lab. So once Annie identified that there was these pyrophosphatases and nucleases involved in the viral life cycle, she wanted to study this in the context of replication. So this was our sort of current model. So what we think that when the microRNA is absent, these pyrophosphatases can remove this triphosphate at the end of the genome. And then the nucleases are free to chew up the viral RNA. So we thought, great, now we know that there's pyrophosphatases involved. Now we know there's a ribonuclease involved. So what we need to do is we'll just knock down the expression of these nucleases and pyrophosphatases. And then hepatitis C virus won't need this microRNA anymore, right? So that's what we did. So we knocked down the pyrophosphatases and the ribonucleases, first in the presence of the microRNA. And you can see that nicely when you knock down these pyrophosphatases and ribonuclease, you see an increase in viral RNA accumulation in the presence of miR-122, suggesting that these do limit HCV replication in the cell. However, when we did this in the absence of microRNA 122, so in miR-122 knockout cells, we saw a little bit of a different picture. So you can see now, like the GNN control, so when you don't knock down anything, in the absence of microRNA 122, hepatitis C virus isn't able to replicate. But if you knock down the pyrophosphatases and ribonuclease, you can see that the RNA is a lot more stable. However, I hope what you'll appreciate from this figure is that this curve goes down, it's not going up. And that means that the virus isn't replicating. The RNA is a little bit more stable, so it's taking longer to decay than it would without these pyrophosphatases and ribonuclease knockdown, but it's not replicating. So this means that microRNA 122 has to have an additional role in the viral life cycle beyond stability. So although we thought we were really smart and figured it all out, we hadn't done that yet. And so we sort of scratched our heads for a while to try to figure out what else microRNA 122 might be doing. And as I told you in the beginning of my talk, RNAs are able to fold into all sorts of complex structures. So we started to think maybe the microRNA could actually be changing the structure of the RNA. And so we decided to use a technique that we use in the lab to study RNA structure. So let me tell you about that and how it works. So we use a technique called selective 2' hydroxyl acylation analyzed by primer extension. And that's a huge mouthful. So we just call it SHAPE for short, or SHAPE analysis. And then the way SHAPE works is that in RNA bases, RNA has a 2' OH group on the base. And when that 2' OH group is base paired with another base or is bound by a protein, that 2' OH group is really static. However, if the RNA is in a single-stranded form, it's more likely to be sampling the environment. So it's kind of waving around like crazy. And when it's waving around like crazy and sampling the environment, we can actually react it with a SHAPE reagent. So we use a SHAPE reagent called NAIN3. And when it reacts with flexible bases, the 2' OH group, it actually forms this 2' O adduct. And this 2' O adduct is important because it sort of serves like a stop sign. So what happens is we treat our RNA with this SHAPE reagent, and at single-stranded positions, it reacts with this SHAPE reagent, causing this little stop sign. And it only reacts in the single-stranded regions, not in the double-stranded regions here. And then what we can do is we can take a DNA oligonucleotide, anneal it to the RNA, and then do a reverse transcription reaction. And when the reverse transcriptase hits one of these stop signs, it falls off, and you get different size fragments depending on how much RNA structure you have. And so then you can take all of these DNA fragments and you can run them out on a gel. And where you see lots of bands, that means that the RNA was unstructured. But when you see an absence of bands, it means the RNA was structured. And from this, we can infer what the actual structure was of the RNA. And so we decided to apply this to our problem and look at the structure of the viral RNA in the presence or absence of the microRNA. So this is work done by Jasmine Shahal, who is the second PhD student in my lab. And so this is the structure of the 5' end of the HCV genome. This is the very end of the viral RNA. And this structure was determined by mutational analysis to see what structures were important for the viral life cycle. And just to remind you, this structure here is the internal ribosomal entry site that we know is needed for the virus to recruit the ribosome to make viral proteins. And so we were pretty sure that this structure was what was functionally important for the HCV life cycle. But what we realized was when we predicted the structure using RNA structure prediction software, the first 117 nucleotides of the genome wasn't actually predicted to form this structure. There was actually 4 other structures that it was predicted to form more stably than this functional conformation. So what we did is we took the viral RNA and we did shape analysis on it to see what it actually looks like in vitro. And when we did that, we noticed that it wasn't actually this functional structure that formed, it was this alternative structure here that I've depicted. And in fact, when we added the microRNA, it actually changed the structure. And the structure changed into this structure, which is actually the functional structure in the viral life cycle. However, we did this all in a test tube with naked microRNA. And as I told you at the beginning of my talk, in our cells, the microRNA comes as part of an Argonaute protein. And so we decided to repeat these experiments, this time in the presence of an Argonaute protein. And here I'm showing you the shape reactivity, but it's sort of a increase or decrease. So it's when the microRNA is there, when the microRNA Argonaute is there minus when it's not there. And so we saw something that we didn't really expect. So what we expected was that when the RNA was in a base pair bound by a protein, the RNA molecules would be blue because they would have a low shape reactivity. And when the RNA was in a loop or in an unstructured part, it would be red. And so we were really surprised by a couple of things we saw when we looked at this. The first was that we saw an increase in shape reactivity here, where we thought that the Argonaute protein was interacting with the viral RNA. And that kind of made us scratch our heads a little bit. The second thing we saw was that there was actually a change in shape reactivity in the iris region, so distant from where we thought the microRNA was interacting with the genome. And if I'm completely honest, this made us really scratch our head for a long time until we reached out to a colleague of ours to ask him if he would help us to model some of these interactions. So we reached out to a colleague at NYU, Hinhark Gann and Chris Gonzalez. And what Hinhark and Chris did was they helped us model the Argonaute proteins on the viral RNA. The viral RNA is shown in teal here, and then the 2 Argonaute proteins are shown here, with the microRNA in red. And what we realized when we did this was that the 2 Argonaute proteins are too physically close together on the viral genome to play nice. So what happens is one of the Argonaute proteins actually releases some of its base pairing interactions in order to accommodate the Argonaute protein at site 1. And so this was really great because it explained that difference in the shape reactivity. But we were still puzzled by the change in shape reactivity in the iris or distant from the HCV site. So we decided to apply this modeling to the whole viral iris in combination with the host cell ribosome. So that's what I'm showing you here. So this is a host cell ribosome in gold and gray here. And then this is the HCV iris in teal. And now I'm showing you the 2 Argonaute proteins in pink and in purple with the microRNAs in orange. And when we did this and we kind of zoomed in on the second Argonaute protein, we realized that the Argonaute protein was actually like sticking its elbow right into the HCV iris, precisely where we saw that change in shape reactivity that puzzled us in our shape data. And so it seems that the Argonaute protein was actually sticking its elbow into the iris, helping to stabilize the iris, we think, helping to promote translation. So if I summarize sort of everything that I've told you so far, this is kind of our model for what we think is happening in the HCV life cycle. So we think when the viral RNA enters into the cell, it's in this conformation. And you can see in this conformation that site 1 of the microRNA binding sites is actually in a base pairing interaction, whereas site 2 is actually single-stranded, so it's available. And so we think what happens is you get recruitment of an Argonaute microRNA 122 complex first to site 2 on the viral genome. And this actually acts as an RNA chaperone or riboswitch to refold the viral RNA. And this actually allows the internal ribosomal entry site to form, including this stem loop 2 structure. And this means that the RNA is now translationally competent, so it can make viral protein. And interestingly, when the microRNA binds here, it actually opens up this region, and now site 1 is accessible. So you can get recruitment of a second Argonaute microRNA 122 complex. And this Argonaute binds to the very 5' terminus of the genome. And as Annie showed us, this actually protects the RNA from being degraded. So it promotes stability of the HCV genome. And interestingly, as I told you, the 2 Argonaute proteins are too physically close on the viral genome to play nice. So what happens is that one Argonaute protein releases some of its base pairing interactions in order to accommodate the Argonaute protein at site 1. And as it does so, it actually leans on and interacts with the viral iris. And we think that this helps stabilize the viral iris, promoting translation of the viral proteins. So if I summarize this part of my talk, I hope what I've convinced you of so far is that a human liver-specific microRNA called microRNA 122 actually facilitates HCV RNA accumulation in hepatocytes or liver cells when HCV infects a patient. And microRNA 122 seems to have at least 3 roles in the viral life cycle. So as I told you, it acts as an RNA chaperone or riboswitch, refolding the viral RNA, allowing the viral iris to form. It also promotes stability of the viral RNA, protecting it from enzymes which would normally degrade the viral RNA. And it also promotes translation through contacts between the Argonaute protein that comes along with the microRNA and the HCV iris. All right, so that's sort of summarizes the first part of my talk. And now I'm going to change gears a little bit and tell you about what we're working on now. So all the stuff I've told you so far is published data. Now I'm going to tell you about some unpublished data. So as I told you in the beginning of our talk, microRNAs usually downregulate gene expression, and they usually cause RNAs to be decayed. What I think I've shown you is that this seems to be the opposite thing happening in this case. And so we started to wonder, how does Hep C actually escape this canonical silencing, even though it's calling RNA silencing complexes to its genome? And so that's kind of what we started to ask a few years ago. And so this is the depiction of the end of the viral genome, and it recruits the microRNA and these Argonaute complexes. And we know in the canonical microRNA silencing pathway, the next thing that happens is recruitment of the TNRC6 protein. And as I told you at the beginning, TNRC6 is kind of the microRNA effector protein. So what it does is it calls deadenylation enzymes and decapping enzymes to remove the poly A tail and the cap of the RNA, and it causes translational inhibition of the RNA. But interestingly, hepatitis C virus, it doesn't have a poly A tail. So it really couldn't care less if you recruit deadenylation enzymes to it. It also doesn't have a canonical cap. It uses an iris to promote translation. So it doesn't really care if TNRC6 recruits decapping enzymes either. So what were we left with? We were left with the idea of translational inhibition. However, there was a lot of disagreement in the microRNA field about how translational inhibition is achieved. And that was until my colleague and collaborator, Ian McRae, found that when TNRC6 is recruited to Argonaute proteins on a target RNA, it causes this phenomenon known as liquid-liquid phase separation, where the TNRC6 protein promotes a droplet formation. Recruits the RNA into a droplet. And this is a really interesting mechanism for how translation is shut off, because these droplets that TNRC6 helps promote on messenger RNAs actually serves as a physical barrier to prevent the ribosome from accessing the RNA. So what is liquid-liquid phase separation? You might not have heard of this. And you know what? I hadn't heard of this until a few years ago. But it's a process by which RNA and protein molecules can come together into a dense phase, which resembles liquid droplets. And this is sort of like vinegar droplets in an oil and vinegar salad dressing. So they can rapidly form. So if you shake up your salad dressing, you see little droplets forming of vinegar. They're characterized by fusion. So two vinegar droplets can fuse together to make a larger droplet. And they're sort of these liquid condensates. And this is common also, you might have seen lava lamps before. So those are also sort of two different phases of liquid droplets. And this is actually the process by which our cells make membraneless organelles. So you might have heard of membraned organelles in our cells, like the nucleus, but actually there's also compartments in our cells that don't actually have a physical membrane around them. They're just liquid compartments. And so this, you might have heard of the nucleolus or stress granules or processing bodies. These are all membraneless organelles. And so we started to think about perhaps this might be a mechanism whereby hepatitis C virus could solve the dilemma of positive sense RNA viruses. So what is the dilemma of positive sense RNA viruses? Well, as I told you, positive sense RNA viruses, they just have a single piece of RNA. And that one piece of RNA has to be a template for all the different parts of the life cycle. So it has to be a template for translation of the viral proteins. It also has to be a template for replication. But interestingly, these two processes actually happen in opposite directions. So the ribosomes bind to the 5' end, and they move from the 5' end to the 3' end to make viral proteins. But the RNA-dependent RNA polymerase, which copies the viral genome for viral genome replication, it starts at the 3' end and travels along the 3' end to the 5' end. And so what this means is that these two processes can't happen simultaneously. Otherwise, you'd get half a viral polyprotein, you get half a viral genome, and the virus would not survive, right? And so in order for viruses to proceed through their life cycles, they actually have to have a mechanism to turn off translation so they can start to replicate. And so what we started to think about was maybe microRNA 122 not only helps HCV turn on translation, but through recruitment of the TNRC6 protein, maybe it also helps Hep C turn it back off. And so this is our working model for how we think that this works. So we think that when the genome enters the cell, it's in this conformation, and then it recruits two Argonaute microRNA 122 complexes. And now the RNA is riboswitched, so it formed the iris. It's stable, so the microRNA promotes its stability, and it's translationally competent, so it promotes translation. And now ribosomes can bind to the viral RNA and make viral proteins. And as the proteins start to accumulate, some of the proteins, one of the viral proteins in particular is really important, and that's the NS5A protein. And what we realized was the NS5A protein had a lot of the same features as the TNRC6 protein, which is a protein that is known to induce phase separation. So we think that NS5A is a phase separation protein as well in the viral life cycle. What we know about NS5A is that it binds to the 3' UTR of the viral genome, the poly-UC tract. And it has these little feet, as I've drawn them here. And these feet are amphipathic helices, which tether it to the ER membrane, or the endoplasmic reticulum membrane. So what we think happens is the NS5A protein starts to accumulate on the poly-UC tract, and it starts to phase separate the viral RNA at the very 3' end of the genome. However, we think it can't condense the whole RNA because there are ribosomes traveling across the RNA making viral proteins. And we think that those ribosomes move the RNA around a lot. And so the virus can't condense the RNA. And so what we think happens next is recruitment of the TNRC6 protein, this time causing condensation of the 5' end of the RNA. And this allows ribosomes to be excluded. And now the NS5A protein is free to condense the whole viral RNA. And as it does so, it actually drags the ER membrane with it and creates the viral replication organelle, which is vesicular in nature in hepatitis C virus. And now the RNA can be replicated in this replication organelle. And when new RNAs come back out, they can either go back through this pathway or they can be packaged into viral particles. So I'm not going to be able to tell you today about what we're doing to test this model, but this is our working model. But I think it has a lot of really interesting implications. And so I want to tell you a little bit about that, or entering into a new phase in virology. So as we started to think about this idea of phase separation in the viral life cycle, we started to think about what the benefits might be of phase separation or biomolecular condensate formation. So these are sometimes called biomolecular condensates. So firstly, this could be really advantageous for a virus because it would help the virus to localize everything into one place. And so it's a way to localize your proteins and your RNA into one location in the cell. The second thing it can do is it can inactivate reactions, right? So as I told you today, it can exclude ribosomes. So it can actually inactivate translation so that replication can occur. It can also cause activation of reactions. So if you concentrate or you crowd factors into one place, you can actually help activate enzymatic reactions. You can also stabilize reactions. So if you're in a more dense environment, you could stabilize a reaction such as polymerization or RNA synthesis. You can also compartmentalize things in these phase-separated compartments. Sometimes the inner phase is more dense, and that sometimes can be where replication is occurring for some viruses. And then transcription and assembly could be happening in the outer layers of the biomolecular condensate. Can also be used to generate force, and this might be a way that some viruses help bud out of the cell. It can also be a selective filter, so allowing some metabolites in while keeping others out. And of course, if the virus can sequester itself into a droplet and keep out things like nucleases or sensors of RNA, it can also serve to protect the viral genome in the cell. And so these are all features that might be helpful for a virus to be able to make this biomolecular condensate or phase separated droplet. And so as we thought about this, we realized that not just hepatitis C virus, but every virus that we looked at actually has a protein or more than one protein that has these features of the ability to phase separate with RNA. And here I'm just showing you some examples from RNA viruses, both positive, negative, and double-strand RNA viruses. But we think that this is a common phenomenon to maybe all viruses on Earth, because the one thing that all viruses on Earth have in common is they have to make RNA. We think that to be able to make RNA in the complex environment of the cytoplasm, perhaps you need phase separation. And so today I've told you about this really interesting interaction between microRNA 122 and hepatitis C virus. I hope I've convinced you that microRNA 122 is really the puppet master of the HCV life cycle. It has at least 4 roles in the HCV life cycle. It acts as an RNA chaperone or a riboswitch to refold the viral RNA into a functional conformation. It provides genome stability, protecting the viral genome from being degraded, and it promotes translation by helping the iris to fold and to be stable. However, after time, it actually also turns off translation and helps the switch from translation to replication. And just by studying this one little microRNA in the hepatitis C virus life cycle, we might have uncovered a common mechanism that maybe all viruses use to establish replication organelles, and we think in some cases also to assemble viral particles. And so that's pretty cool that we can learn something about all viruses by studying one virus. So with that, I'll make my acknowledgements. I want to thank all my lab. They're the ones that really did the work. So Annie and Jasmine were the ones that started this project. Sophie and Mary Lynn also carried this on. And recently, Carolina and Manolia are working on this project. I have lots of collaborators in the States and in Canada that really helped us push these projects forward. And of course, I'd love to thank my funding sources for allowing us to continue this work. And I'd really love to take your questions today about anything that you heard today. So thank you.
Excellent. Thank you, Selena. That was really great. Yeah, thanks everyone for your attention, and we welcome any questions either in the chat. Feel free to type in your questions, or you can raise your hand and ask your question using your mic. So we'll let you get us started. And maybe while folks are thinking of their question, I thought maybe I'll ask. And Oh, let's go to Tyler. Go ahead, Tyler.
Uh, hi, hi, Professor. Thank you a lot for your presentation. I have one question I sort of think of about sort of viruses. I want to
Yeah.
roughly ask how far are we between our current technique and understanding to the state, possible, maybe may not possible state of zero virus and zero diseases? Is it is it even possible?
So I would say no. And I think if I'm honest, that's actually one of the other reasons that I thought that studying viruses would be such a lucrative career, because I think there's always going to be disease and there's always going to be viruses. And so there's always going to be a need for someone to study them and understand them. And so for me, that was something that was really attractive as an undergraduate student, because I thought it was sort of job security for life.
Yeah, thanks, Tyler. There's a question in the chat. Jazzy's asking, I don't know if Selena, if you can see the chat, but I can read them out.
Yeah, great.
Yeah, Jazzy's asking, do RNA viruses have a biological advantage compared to DNA viruses? And also, if you, they're asking, have you ever played Plague Inc? And do you know, or do you think the game is accurate to real world diseases and spread?
Okay, so to answer the first question, I think that it's hard to say whether they have a competitive advantage. One advantage over in RNA viruses over DNA viruses is that they have a really high mutation rate. So they actually make a lot more mistakes, and so they can evolve in a lot of cases a lot faster than DNA viruses. And in fact, most of the emerging zoonoses and emerging viruses that affect humans are RNA viruses. So something between 25% and 44% of all emerging infectious diseases are RNA viruses, in fact. However, DNA viruses also sort of have a bit of an advantage maybe over RNA viruses, because in a lot of cases, they can use a lot of the host cell machinery. So our cells have DNA-dependent DNA polymerases and DNA-dependent RNA polymerases, but our cells don't actually have RNA-dependent RNA polymerases. So RNA viruses have to bring those with them, whereas DNA viruses might take advantage of some of our machinery in the host. So it's harder to say if there's like an advantage of RNA versus DNA viruses, but certainly RNA viruses seem to be more potent threats, I would say, for emerging viruses today.
Yeah,
And
great.
I haven't played Plague Inc. I have heard of it, but I haven't played it, so I can't really comment on how accurate I think it is.
I'll have to check that out. Is that an app like for a phone or something, or?
I think it's like a game that you
Yeah,
can play. Yeah, like
yeah.
online. Yeah.
I'll have to look at that. Let's go to Bella. Go ahead.
Hello, thank you for your talk. It's very informative. I was just wondering, how is it discovered that the miRNA 122 binds to the 5' end, and why does it do this?
Yeah. So good question. So it was discovered by looking at the complementarity. So basically, in the lab that I did my postdoc in, they discovered it a few years before I arrived in that lab at Stanford. And what they noticed was that there were sequences in the Hep C genome that really that were complementary to the microRNA, and they were super highly conserved. So like 100% of all HCVs all around the world, even though there's 7 different types of genotypes, they all had this conserved microRNA binding sites. And so what they did was they mutated the site in the virus, and then they made a compensatory mutation in the microRNA to show that actually when you mutate it, you lose the interaction. When you make a compensatory mutation in the microRNA, you gain back the interaction. And so there's a genetic interaction between the microRNA and the virus. And then why does it bind in the 5' UTR? So I think this is a question that's plagued us for a while. So I think for 2 reasons. So as I talked about today, the microRNA actually binds to the very terminus of the viral genome, and that means it can protect against nucleases and pyrophosphatases, which can decay the RNA. So I think that helps because it means that the 5' end is not available for those cellular decay enzymes. I think the other reason is really for the for the sort of latter part of my talk, and it's because it's right next to the internal ribosomal entry site, that structure that forms that can recruit the ribosome. And I think that what happens is that if you phase separate right next to that structure, then you hide that structure from the ribosome, and the ribosome can't access it. So I think that that's probably why these interactions are occurring in the very 5' end of the viral RNA rather than the 3' end of the viral RNA.
Great question, Bella. Yeah. I actually had a follow-up. What initiates phase separation in the first place? Like what triggers it?
Yeah, so that's a great question and something that I'm still trying to figure out myself, if I'm honest. But there's some really interesting information about proteins which tend to undergo phase separation. So they tend to have these large intrinsically disordered domains, and that means that they don't have a fixed structure. So they kind of can sample a lot of different structures. They tend to have low complexity sequences that are enriched in certain amino acids that are kind of like sticky, is what I would call them. They tend to be RNA binding proteins, and they tend to be proteins which can form dimers or higher ordered oligomers. And so what we think happens is that you get a lot of weak sort of sticky interactions, and those sticky interactions promote sort of liquid condensation. So you get lots of interactions between the proteins and between the RNA, and this tends to make a liquid. But if those interactions are too strong, it moves it towards an aggregate or a solid. And so it's sort of the features of some of these proteins that bind to the RNA that help make these sort of liquid condensates. But we don't know all the precise details of how this happens. And that's something that we're trying to really work out in the lab.
So you can imagine that it's kind of like transient and dynamic, keeping it sort of fluid. I liked your model of perhaps it being important in like force generation and like pushing on membranes or pushing things, changing shapes inside the cell, which is pretty neat. Okay. Any other questions to come through on the chat, or please feel free to raise your hand if you have a question.
Sorry, I just have a question.
Go for it, David. Yeah.
Thanks so much for Dr. Sagan giving this presentation. And since you just talked about micro ribosome 12.2 and HCV life cycle, like, can you please explain what kind of beneficial effects can we have? Because you do mention at the beginning of the presentation that the HCV is like causing 40 to 60% of chronic liver disease. Like if the micro 122 is gonna like lower the rate, or it's gonna like suppress the expression.
Yeah, so basically what happens if you use an antisense molecule against the microRNA, it sequesters it, kind of hides it from the virus, which really wants to use the microRNA. And because the virus can't use the microRNA, it can't accumulate in cells because it has a really hard time translating and then switching from translation to replication. So what ends up happening is it sort of gets lost in translation, is sort of how we think about it. And so the microRNA, if you sequester it, you can prevent it from getting to hep C. And that's what happened in these patients when they were treated with an antisense microRNA 122 inhibitor. Does that answer your question?
Yes. I think probably like just the inhibiting of the accumulating will not like have a like obvious disease display.
Yeah. So I mean, if you can clear the patient of hepatitis C virus, then they're not going to get any disease, right? So if you can clear the disease, then they shouldn't get liver cancer. Their liver shouldn't sort of fall apart over time. So hepatitis C virus causes like advanced liver disease and causes steatosis, which is sort of fatty liver, and can cause scar tissue in the liver and can eventually lead to a need for a liver transplant or liver cancer. And so if you can stop HCV in its tracks, then you can stop some of those things from happening to patients.
Perfect. Thanks so much.
Yeah.
Do you foresee there being any sort of viable antivirals based on the phase separation discovery and how universal it is?
Yeah, so I think that's really interesting. It's something that we think about a lot because there are inhibitors that we use in the clinic today to treat hepatitis C virus. And in fact, they target the NS5A protein, but no one knows how they work. And so one of our hypotheses is that maybe they're actually phase separation inhibitors. And we actually already have antivirals that we use in human patients that are phase separation inhibitors today. If that's true, and that's something that we're trying to resolve in my research lab, then there is a lot of precedence for anti-phase separation inhibitors as antivirals. And I think that there's a way for us to target sort of the flavor of phase separation that viruses undergo in order to set up their replication organelles. So I think there would be a way for us to target this in a virus-specific manner.
Yeah,
And that's the hope.
Yeah, that's so interesting. Because yeah, I was wondering that like would an inhibitor have any sort of negative impacts on phase separation that's normal for the host cell? Or is there
Yeah,
something, like you said, a unique viral flavor of that type of condensate?
yeah, so I think there are like these very general phase separation inhibitors. I don't think that'll do it because I think you're probably phase separating lots of different compartments in the cell to make other reactions happen. But I think that if we really target these viral proteins, which are mediating the phase separation, then I think that we could sort of target a unique flavor of phase separation that's required for the virus.
Great, thanks very much. Any other questions? Could be about the science or viruses, or perhaps Dr. Sagan's path in science or passion. I have another question that might be of interest to folks. I wondered if you could just describe kind of like what a day in the life in your lab looks like. What do the experiments look like that you would typically use to test some of these ideas and hypotheses?
Yeah, so we use lots of different techniques in the lab, but luciferase assay is one of our favorites. We really like to use reporter RNAs. The reason we like to do that is because luciferase assay is actually like quite quick to do. So we can very quickly report on what happens to the virus. We also do a lot of genetic manipulation. So we have copies of the viral RNA in a plasmid, and then we can make mutations in it, and then make those mutant RNA and introduce those into cells. So that's something that we use a lot because when we want to sort of make mutations and see what happens to the virus. So we do that quite a bit. As I said, we do a lot of shape analysis, which is an RNA structure process, RNA structure sort of probing process. And then we do some pretty standard molecular biology assays. So we do Western blots to look at protein levels. We do Northern blots or quantitative reverse transcription PCR to look at viral RNA levels. And we do a lot of imaging as well. So we use fluorescence in situ hybridization to stain for the viral RNA in cells and to stain for viral proteins as well. And then, of course, the classical assay that we do a lot in the lab is the plaque assay, which is a way to measure infectious viral particles, and a focus forming unit assay because hepatitis C virus doesn't form plaques. So in that case, what we do is we stain cells and we look for foci of infected cells to quantify infectious viral particles. So those are some of the classic techniques that we use in the lab.
And I wondered, maybe you could comment on our students might be interested in like, how do you work with viruses? Like, what does that look like to make sure that it's like safe for folks to do their experiments? Like, what does that look like in your lab?
Yeah, so we run a BSL 2+ or biosafety level 2+ lab. And so what that means is that we have a BSL 2 lab with BSL 3 precautions. So we have some extra precautions because the viruses that we study are bloodborne pathogens. And so what those extra precautions are is that we don't use glass, or we don't use any needles, anything that could cause a cut or abrasion. And we perform all of our experiments in a biological safety cabinet. So anytime we use virus, it's within containment. Unfortunately, the viruses that we study don't have vaccines against them. So we can't have any vaccine against the virus, but certainly some of them have direct acting antivirals that could treat if there was an issue. However, what we do is we work very safely. So we have very standard safety protocols for how to handle viruses and how to maintain safety. And of course, we inactivate viruses at the end of experiments so that we don't have anything that's infectious at the end of the day. So all of that's done in biosafety level 2+ cell culture facility here in my lab at UBC.
Great, thank you. Yeah, Caitlin, go ahead.
Sorry, just gonna lower my hand here. So we have some of our students, and maybe they're a bit shy to ask a question, but some of our students are researching for their term paper topic, the RNA world hypothesis. And so it's up to them to search for evidence sort of for or against this. But I wondered if you might have anything like that you want to share your opinion or advice for students who might be researching that topic.
Well, one, like, cool topic, awesome job, because RNA is really cool, and the RNA world hypothesis is really fascinating. I'm trying to think of like specific advice, and like nothing's like really popping to mind at the moment, but I just think it's the idea of self-replicating RNAs. So there's a lot of research on this topic today. So the RNAs that can replicate themselves or undergo enzymatic reactions, really being the precursors to sort of life is really a fascinating topic. And I really do think RNA sort of was here first before DNA or protein. So I think that will be really interesting to uncover over time. The other thing I think about is, viruses. Also, one of the hypotheses is that viruses predated cellular life. There's lots of hypotheses about viruses, where viruses came from, but that's one of them. And so, even though I'm studying RNA virus interactions today, I do think about whether RNA or viruses or both were sort of precursors to life on Earth. So certainly an interesting topic. One additional thing that I'll put out there, which I think is really interesting, is these proteins, which actually undergo phase separation, they are enriched in these low complexity domains. And these low complexity domains are actually of amino acids, which were the first amino acids in early Earth. And I think that's because things needed to be sticky for reactions to happen, right? And I think that might have some interesting thoughts about how things happened in primordial Earth, right? Because you need things to stick together for long enough for reactions to happen. And so these sort of sticky amino acids were some of the first amino acids that appeared on primordial Earth. And so there might be something to do with this phase separation idea also allowing life to occur.
Oh, that's really cool. Thanks.
Yeah, no problem.
Excellent. Okay. Well, if there aren't any other questions, maybe we'll end it there. But I want everyone to join me in thanking Dr. Sagan for a fascinating seminar today. Thank you.
Yeah, thanks so much for having me.
Yeah, it was really great.
Great.
Actually, perfect timing because we're just coming up to 1:50 when people might need to go to their next class. I'm just going to stop the recording now.
Perfect.